Welcome back. This video is on the double helix structure of DNA. And the learning goals of this session are 1. Recognize the structural components of a nucleotide. 2. Name the 4 nitrogenous bases in DNA and RNA. 3. Outline how the DNA nucleotides are linked together into a polynucleotide chain. 4. Understand the Chargaff's rule and its implication in DNA structure. 5. Describe the salient features of the double helix model of DNA. And 6. Draw a schematic illustration of DNA dinucleotide chains showing the correct polarity. The discovery of the double helix structure of DNA was the collaborative research and contributions from lots of scientists. Although many people don't realize it, the first piece of the puzzle was solved by a Swiss physician named Friedrich Miescher in the year 1869 when he first isolated and discovered nuclein, which is now known as nucleic acid, from the nucleus of pus cells or WBC cells. After him, an American biochemist named Phoebus Levine in the year 1919 discovered the chemical components of nucleic acids, and he also proposed the polynucleotide model in order to explain with the binding of each nucleotide to form a polynucleotide chain. An Austrian biochemist named Erwin Chargaff expanded on Levine's work by uncovering additional details. From his studies, he finally came out with 2 major conclusions in the year 1950. The first conclusion was that the nucleotide composition of DNA varies among species. In other words, the same nucleotides do not repeat in the same order in every organism. And secondly, that the amount of adenines A always equals the amount of thymines T, and the amount of guanines G always equals the amount of cytosines C in all DNA, no matter which organism it came from. And this second conclusion is now known as the Chargaff's rule, where the amount of adenine is always equal to the amount of thymine and the amount of guanine is always equal to the amount of cytosine, and vice versa in all DNA. Or in another way, it can be also expressed as the amount of purines is always equal to the amount of pyrimidines in all DNA. Another landmark contribution came from 2 English chemists named Rosalind Franklin and Maurice Wilkins, who were working in the same lab. They used a technique known as X-ray crystallography, which is a technique in which chemical molecules, and now in this case DNA, was exposed to high beams of X-rays. And thereby they were able to produce X-ray diffraction photographs of DNA to study the molecular structure. Now here is one of Franklin's X-ray photographs of DNA, which is the famous photo 51, where we can beautifully see the X-shaped pattern of dark spots. While Franklin was trying to confirm with the molecular structure of DNA, her colleague Wilkins shared her photograph with Watson without her knowledge. And that was it. Watson and Crick was able to immediately confirm with the double helical structure of DNA, and that's how they succeeded in proposing the double helix model of DNA structure in the year 1953. In fact, Watson, Crick and Wilkins were awarded with the Nobel Prize in 1962 for the discovery of DNA structure. Sadly, Rosalind Franklin's contribution was largely ignored despite the fact that her famous photo 51 was instrumental in the discovery of double helix model. As proposed by Levine, each nucleotide is composed of 3 elements, which are a pentose sugar, a phosphate group and a nitrogen base. Pentose sugar refers to a sugar containing 5 carbon atom, which is ribose in RNA and deoxyribose in DNA. Ribose and deoxyribose differ in one functional group. Now look at the illustrations here. In RNA, pentose sugar is oxygenated, and in DNA, pentose sugar is deoxygenated. Which is to say that in RNA, hydroxyl group is present at the 2nd prime carbon position of the ribose sugar, whereas in DNA, oxygen is removed and only hydrogen is present at the 2nd prime carbon position of the deoxyribose sugar. Phosphate group is going to be imparting with the negative charge to the DNA due to the presence of highly ionized oxygen atoms in the phosphate group. And nitrogen bases can be broadly categorized into two classes, which are of the double-ringed purines and single-ringed pyrimidines. Purines include adenine and guanine, and pyrimidines include cytosine, thymine, and uracil. Both DNA and RNA are going to be having 4 nucleotides each. In DNA, it has adenine, guanine, cytosine, and thymine, while in RNA, it has adenine, guanine, cytosine, and uracil. So the difference between DNA and RNA in terms of nitrogen base is, in RNA, thymine is replaced by uracil. The next question is, how do we have the 3 elements in a nucleotide linked together? To give you an idea on the structure of a nucleotide, I'm going to be using 3 cutout shapes here. This pentagon is representing a pentose sugar, and you can assume this to be a deoxyribose sugar in the case of DNA. This rectangle is representing a nitrogen base, and the circle is representing a phosphate group. Chemically, in each nucleotide, a pentose sugar is covalently linked with a nitrogen base at its first carbon position and a phosphate group at its 5th carbon position. And the covalent bond formed between the first carbon of the pentose sugar to that of the nitrogen base is called as N-glycosidic bond. And the covalent bond between the 5th carbon of the pentose sugar and the phosphate group is the phosphoester bond. In a nucleotide, covalent bonds are formed through condensation reactions. And depending on the type of nitrogen base in a nucleotide, nucleotides can further be categorized into 5 broad types, which are 1. Adenosine monophosphate. In this type of nucleotide, the nitrogen base which is forming the nucleotide is adenine, hence the name adenosine monophosphate. The second type is guanosine monophosphate, where the nitrogen base is guanine. The third type is cytidine monophosphate, in which the nitrogen base is cytosine. The fourth type is thymidine monophosphate, in which the nitrogen base is thymine. And the last type is uridine monophosphate, which is only found in RNA. And here the nitrogen base is uracil. So in DNA, the 4 types of nucleotides are adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, and thymidine monophosphate. While in RNA, the 4 nucleotides are adenosine monophosphate, guanosine monophosphate, cytidine monophosphate, and uridine monophosphate. If the pentose sugar attaches with only a nitrogen base, then such a molecule is referred to as a nucleoside. So you may want to note the difference between the structure of a nucleoside and the structure of a nucleotide. A nucleoside is consisting only of a sugar and nitrogen base, whereas in a nucleotide, we have a sugar, nitrogen base, and a phosphate group. So a nucleoside is lacking in the phosphate group, whereas a nucleotide has the presence of a phosphate group. So basically, nucleoside is formed through the hydrolysis of nucleotide. When the phosphate is removed from the nucleotide, then it becomes a nucleoside. And just like a nucleotide, nucleosides can also be classified into 5 basic types, depending on which nitrogen base is attaching with the sugar. Now, if thymine is attaching with the sugar, then the nucleoside is called as thymidine. If adenine is attaching with the sugar, then the nucleoside is adenosine. If guanine is attaching with the sugar, then the nucleoside is guanosine. If cytosine is attaching with the sugar, then the nucleoside is cytidine. And if uracil is attaching with the sugar, then the nucleoside is uridine. So in this way, we have 5 different types of nucleosides. And nucleosides, as I said earlier, are basically lacking in the phosphate. So nucleoside is nucleotide minus phosphate, whereas a nucleotide can be formed by phosphorylation of a nucleoside. That is, addition of a phosphate to a nucleoside will result in a nucleotide. DNA and RNA are complex polynucleotides in which a chain of nucleotides are linked together. To show you how the nucleotides are linked, I have displayed structure of a dinucleotide. Over here, we have 2 nucleotides which are linked together by means of a covalent bond called as phosphodiester bond. So what is this phosphodiester bond? Now this is a bond which is formed when phosphate group attached to the 5' carbon of the sugar in one nucleotide forms a second ester bond with the 3' carbon of the sugar in another nucleotide. Since the phosphate group forms two ester bonds between the nucleotides, such a bond is referred to as phosphodiester bond. And in this way, you can add more nucleotides to build the DNA chain. In a human diploid cell, we have over 6 billion nucleotides linked together by phosphodiester bonds. Let me now explain the details of the double helix model of DNA proposed by Watson and Crick in the year 1953. And in this context, what I want you to keep in mind is that there are several forms of DNA, and Watson and Crick's double helix model describes the structure of the most common form of DNA, which is the bDNA. Now, one of the apparent salient features of a bDNA or the DNA structure is that DNA is a double-stranded helix, which means to say that there are two strands or there are two polynucleotide chains in a DNA, and these two polynucleotide chains are twisted along a common axis. The exterior or the backbones of the two strands are formed by alternating sugar-phosphate-sugar chains of the polynucleotide, while the nitrogenous base pairs are projecting towards the inside of the double helix. Another important feature is that the two strands are anti-parallel. To understand this, you need to know the polarity of the DNA strand. So there are two polarities which are marked as 5' end and 3' end in each DNA strand. The end with the free phosphate group at the 5' carbon position of the sugar is referred to as the 5' end, and the end with the free hydroxyl group at the 3' carbon position of the sugar is referred to as 3' end. Now, when we look into the polarity of the two strands on its either ends, what we see is that the 5' end of one strand aligns with the 3' end of the other strand, and vice versa. That is, the 3' end of one strand aligns with the 5' end of the other strand. So this simply means to say that the two strands run in opposite direction. The third salient feature of a bDNA is that the two strands of the DNA are twisted in a right-handed fashion along a common axis. And in such a right-handed helix, the pitch of the helix, which refers to actually the height of one turn when the strand makes a complete 360-degree turn. So the pitch of the helix or the height of each turn is found to be 3.4 nanometer, and the distance between two consecutive base pairs was found to be 0.34 nanometer. Therefore, the number of base pairs in each turn is estimated to be about 10 base pairs in bDNA. The next salient feature is that the two strands of the helix are held together by hydrogen bonds between complementary nitrogen bases. So the base pairing between two strands of the DNA aren't any combination of bases. Instead, adenine selectively pairs with thymines with two hydrogen bonds, and guanine selectively pairs with cytosines with 3 hydrogen bonds, and vice versa. And these ATGC pairings are referred to as complementary base pairing. Chargaff's work has been critical in determining with the complementary base pairing principle. Chargaff proposed that the amount of adenine always equals the amount of thymine, and the amount of guanine always equals the amount of cytosine in all DNA. And consistent with this proposal, adenine pairs with thymine and guanine pairs with cytosine, or thymine pairs with adenine and cytosine pairs with guanine, thereby keeping with the consistent proportion of purines to the proportion of pyrimidines in a DNA. Now, because a larger purine, such as adenine and guanine, always pairs with a smaller pyrimidine, such as thymine and cytosine, so this kind of complementary base pairing would always allow for a uniform distance between the two strands of the DNA, or this would allow for a uniform diameter all along the length of the double helix. The uniform diameter of the double helix has been found to be 2 nanometer. Stability of the double helix DNA structure is largely dependent on the presence of hundreds to thousands of hydrogen bonds between base pairs. And since there are 3 hydrogen bonds between GC base pairs and only 2 hydrogen bonds between AT base pairs, so a DNA molecule having higher proportion of GC would be showing greater stability over a DNA molecule having higher proportion of AT base pairs. In addition to the many hydrogen bonds between base pairs that confers stability to the double helix structure of the DNA, the base pairs arrangement is such that they are stacked one on top of each other, and this kind of the stacking of the base pair also confers stability to the double helix structure. Well, the discovery of the double helix structure of the DNA set the stage for understanding how genetic information is stored and transmitted from one generation to the next generation. And since 1953, scientists and researchers have made unimaginable progress in the field of medicine, agriculture, evolution, DNA fingerprinting, human genome project, and gene therapy because of the landmark discovery of the double helix structure of DNA. Finally, here's a schematic illustration of a double-stranded dinucleotide structure. And with the help of this schematic illustration, let us try to review the structure of nucleotide and the structure of DNA. So let's first try to recall the structure of a nucleotide. We learned that each nucleotide is consisting of 3 elements: pentose sugar, a nitrogen base, and a phosphate group. And we also learned that nitrogen bases are broadly of 2 types: the bigger purines, which are adenine and guanine, and the smaller pyrimidines, which are cytosine and thymine in DNA. We also know that nitrogen bases are covalently bonded with the first carbon of the sugar by means of an N-glycosidic bond, and the phosphate group is covalently bonded to the 5th carbon of the pentose sugar by a phosphoester bond. Now, the nucleotides in the DNA are linked together by means of another covalent bond which is known as phosphodiester bond. So this is one polynucleotide chain, and here is the opposite or the complementary polynucleotide chain. So DNA is double-stranded. And in a DNA structure, what we learned was the backbone of the DNA is phosphate-sugar chain. So you can see phosphate-sugar chain, which are the backbones of the 2 strands of the DNA. And the nitrogenous base pairs are projecting towards the center or core of interior of the double helix. Now, if you look into the base pairing of the nitrogen bases, we learned that base pairing has to be always between complementary bases. So adenine should always be pairing with thymine on the opposite strand, and cytosine should always be pairing with guanine on the opposite strand. Or you can say guanine pairs with cytosine and thymine pairs with adenine with the help of hydrogen bonds. Now, if you look into the direction of the 2 strands, so as we know, each strand has 2 polarity. It has a 5' end and a 3' end. So in this strand, in this left side strand, here is the 5' end because you have the free phosphate group of the 5' carbon sugar at this end, and the bottom end is the 3' end because here we have the free hydroxyl group of the 3' carbon sugar. Whereas in the right side strand, we have 3' end which is at the top and 5' end is at the bottom. This is the 3' end of this strand because here we have the free hydroxyl group of the 3' carbon sugar, whereas this is the 5' end of this strand because here we have the free phosphate group of the 5' sugar. And if you look into the direction of the 2 strands, we see that in this left side strand, 3' to 5' is running in the upward direction, whereas in the right side or the opposite strand, what we see is if you go from 3' to 5' direction, then the strand runs from the top to the bottom. So it is running in the opposite direction or in the downward direction. And that's the reason why we said that the 2 strands of the DNA are anti-parallel. They run in opposite direction. So this is how we can be figuring out all the different components of the nucleotides and the how the polynucleotide chains are constructed and how we have the two strands of the polynucleotide chains in the DNA held together by means of hydrogen bonds between the complementary base pairs. With this, I end my lesson on the double helix structure of DNA, and I'll catch up with my next video lesson on DNA packaging.